I'm Dr. Edwin George, and this presentation is on interactions between genetics, intracellular environment, and alpha-synuclein. What does it mean for multiple system atrophy? And it's sort of a update continuation on what I spoke on last year, when I was very excited that we might be coming down to some better types of diagnostic tests. So I'll begin with a disclaimer. I worked for decades for Wayne State University as a clinician in the medical school and a researcher. I retired from Wayne in the last year, and I now work for the FDA, reviewing new drugs that are being proposed. But this talk has nothing to do with what I do for the FDA. And so this is just my views based on my current reading of the literature. I also have nothing to disclose. So as I mentioned last year, it looked a lot simpler. Lewy body and other synuclein pathologic changes spread through the brain from brain cell to brain cell. A misfolded protein, alpha-synuclein, was the agent. It could fold and aggregate in different ways to cause different diseases, and it looked like we would soon have tests that could actually detect different strains of misfolded alpha-synuclein, and hopefully in that way distinguish Parkinson's disease from multiple system atrophy, from dementia with Lewy bodies, and so forth. But it was too good to be true. So these are a few slides from last year just to remind you about Lewy body pathology in Parkinson's disease. This was the original study by Brock and co-workers, where they showed that the changes of alpha-synuclein-based Lewy bodies in Parkinson's began in the brainstem and then gradually spread up and down the brainstem and throughout the brain progressively. And similar sort of spread has been demonstrated in other synucleinopathies, including multiple system atrophy. And these are some of the papers over the years showing that alpha-synuclein fibrils could induce the aggregates that cause the Lewy body pathology and other types of synucleinopathy pathology, and that this actually spread from cell to cell of the pathogenic protein in these various neurodegenerative diseases. And this is the concept up at the top of the slide here. You see this is an alpha-synuclein polypeptide, a piece of protein, unfolded, a string of amino acids, and it can fold up in a number of different ways, represented just below that by these different geometric shapes. And some of these shapes can stack together, and in fact, they induce other unfolded alpha-synuclein protein chains to fold up in a similar fashion and add to their stack, so that down at the bottom of the screen, you begin to build these different aggregates and fibrils made up of alpha-synuclein molecules. And the idea was that these forms that aggregated like this gave rise to different diseases. So for instance, this one over here might give rise to Parkinson's disease, whereas this one over here might give rise to multiple system atrophy. And so these are the synucleinopathies: Parkinson's disease, of course, Lewy body dementia, I mentioned, multiple system atrophy, which we're all interested in this conference, and also pure autonomic failure. And again, from last year, these are seed amplification assays where bits of tissue homogenized from, in this case, autopsy brains of people. So at the autopsy, we knew for sure who had multiple system atrophy, who had Parkinson's disease, who didn't have either of those, so they were healthy controls. And you could cause the proteins to aggregate. You provide alpha-synuclein in a solution, but put a little bit of purified homogenized brain tissue in to induce the type of aggregation that you want to have. So if you had this kind of thing in there, it would generate more of the same because of the seeding as the aggregation of the protein in solution. And using a fluorescent probe that would bind to this, you could see the aggregates forming in the solution, and you could tell them apart. And you could tell them apart both by how much fluorescence they bound, and also by the speed at which the aggregation took place in the solution. And here is another one showing a similar experiment, in this case using dementia with Lewy bodies and Parkinson's disease, and being able to distinguish them. So it looked very promising that soon we'd be able to use this kind of test to say, okay, you have multiple system atrophy, you don't have Parkinson's disease, which is often a big question. And people were already doing tests like this in spinal fluid, and it's available now. And again, it does pretty well. You see all these high statistics for specificity and pretty good sensitivity. And this test was just, and still is, just determining that there are, in fact, aggregates in the spinal fluid that can seed and produce protein aggregation in solution that's an alpha-synuclein aggregate, but not what particular alpha-synuclein aggregate it is. And we also have tests where you can use, again, fluorescent probes that bind, and in this case, glow green, where they've bound to alpha-synuclein aggregates that are deposited, in this case, in a skin biopsy, showing that you can see the aggregate protein present in the skin, and some suggestion that maybe exactly how deep in the skin had some reflection on exactly what kind of underlying disease you had. And so people started asking me, well, will my synuclein test tell me which disease I have? Unfortunately, as research has progressed, we've discovered a lot of interesting things, but the potential to have a simple test has become much more complicated. So this is just to remind you, and we'll come back to it throughout and at the end as I talk, that there are genetic factors that can change the risk of having, in this case, multiple system atrophy, though many of these are also associated with regular Parkinson's. And there are environmental factors that can affect your risk of developing these diseases, and probably affect your risk of actually beginning to develop aggregation of alpha-synuclein in your body. Now, the thing about alpha-synuclein misfolding is it can actually misfold in many ways. And as we saw in that slide with the little geometric things, there were a whole bunch of different geometric shapes, only two of which were aggregating to cause a particular type of aggregate that we were saying, well, this could be related to one disease and that could be related to another. And in fact, the strains, the different forms of aggregation that are disease associated are really quite few. But the protein itself not only can misfold in many ways, but a lot of the different ways can form aggregates. And there are a lot of variations in aggregates that can occur. Nonetheless, only a few seem to be specifically associated with specific diseases. So this is another slide sort of related to the one I showed you early with the geometric shapes. Now we're using Lego blocks over here to show different ways that subunits can aggregate to form different types of aggregates. And over here again, we have a diagram of the amino acid chain that makes up a molecule of alpha-synuclein. And here there's just one molecule, and this is one way it can fold up. And it could then, each of these could be a subunit, maybe make something relatively simple like this. Each of these others are ways that two molecules of alpha-synuclein can fold up and join together. And then they could be subunits for some of these more complicated assemblies represented by the Lego blocks. So the question is, how do you end up with one particular strain of alpha-synuclein aggregate that's causing the disease in question? And the aggregation has to be triggered somewhere. So over here, we're envisioning a cell in which aggregation of alpha-synuclein becomes triggered, which could happen for a variety of reasons, and could actually not only form a bunch of different small so-called oligomers, the subunits of the bigger fibrillar chain aggregates, but also could form several different types of bigger fibrillar aggregates. But the disease doesn't stay in one cell. It passes from cell to cell. And there are factors that can affect what kind of aggregate gets passed from cell to cell and forms in different cells. So it turns out, and this occurs in those seeding amplification assays that I showed you earlier, the conditions in the solution make a big difference in how readily different types of aggregates will form. So some people who initially tried doing the test, for instance, said, no, we can't get any aggregation from seeding with multiple system atrophy samples. And other people got aggregation that was pretty clear. And it turns out that what the conditions around the protein are affect what kind of aggregation and how readily it can aggregate. And so this cell not only could give rise to several potential ways of forming fibrils, but it could in fact have its own subset of the potential fibrils that could form. But then it has to pass it to the next cell. And actually the mechanisms by which these little protein aggregates and fibrils can get from cell to cell can be selective for some types of aggregates versus others. And then that cell has its own intracellular milieu that may affect what kind of aggregation gets amplified within that cell. And then you have another step of transmission or more, multiple steps, where each time the mechanism of transfer and the milieu in the receiving cell and its susceptibility to amplifying these aggregates may be quite different. And so, as you can see here, these authors have hypothesized that if the trigger cell sends things this way, you end up with this kind of strain. And if the trigger cell sends things this way, you end up with that kind of strain, with the different steps of cell-to-cell transmission focusing down onto exactly what the predominant strain is going to be, which will in turn determine whether there's disease and perhaps what kind of disease, whether it's, say, Parkinson's versus dementia with Lewy bodies versus multiple system atrophy. And then again, this is theoretical to go with it, but again, using fluorescent probes to look at the conformational landscape. And that's one of the newer things that's very exciting. We have newer probes that bind to the aggregated proteins. And these probes fluoresce to give you these different fluorescence and different wavelengths. And you have to make ones that are selective to bind to alpha-synuclein. But then depending on how the probe molecule gets bent and the local environment that it finds itself in on the surface of the aggregate, it can change its fluorescence wavelength a little. Now, wavelength ultimately, invisible light is what gives us color. So you can think of these as little dyes that you can put on living cells that get in there and bind to the misfolded alpha-synuclein aggregates and may change color according to exactly how the misfolding has taken place in that particular piece of protein. Now, the shifts in wavelength are very small, and you need very good equipment to detect the change in wavelengths, but nonetheless, it can be done. And so conceivably, if you looked at a trigger cell, you might find this smear of differently folded alpha-synuclein. And as it moves on to the next cell, the selective properties of how it gets transferred, whether the cell will receive it or not, whether it can amplify it or not, what the conditions are in that cell for actually amplifying, forming more aggregates, narrows down the variety until you end up with a single strain or minor variations in a single strain that might cause a specific disease. And again, as we saw in the previous slide, you can go to cell A or you can go to cell B, and different fluorescence according to how those aggregates are folded. So one of the things, this says the infection hypothesis, there's long been the theory that the Parkinsonian diseases could be triggered by some forms of infection. And it turns out that some forms of infection actually do stimulate more alpha-synuclein production in different types of cells, and more conditions within the cells that may favor aggregation of one sort or another. And so in this artist's conception, we have a variety of infectious agents, including bacteria and viruses, that can infect different areas where we suspect that the diseases may start. The olfactory tract can be involved in Parkinson's disease. The gut can be involved in most of these diseases. The bladder could be involved. And so the trigger cells could be hanging out in any of these locations and become triggered by the effects of the infectious agent, but also modified by the genetic background. Some of these cells may be different with different genes that changes their tendency to form aggregates, and perhaps what kind of aggregates they form. The presence of environmental factors in these places could also have an effect on how likely aggregates are to form and what kinds are likely to form. And finally, the host immune response to the infection may have effects on the possible triggering of alpha-synuclein seeding into aggregations. And so here, the idea, maybe through the olfactory tract and then the cell-to-cell transmission sort of thing that the previous authors were suggesting, will bring you down to Parkinson's disease or dementia with Lewy body disease in the brain. But a different infection and a different route of transmission may end up favoring a protein misfolding that causes multiple system atrophy in the brain. Well, you can take this a step further. So this is a different paper that looked at some of these data and suggested that there's differences in the strains and in the pathology as things spread. And so you can have things that start actually with a trigger cell in the body, and here it says the gut, so here's the number 1. and you make some alpha-synuclein aggregates of a particular strain, and it gets transferred up and also out into the nervous system connected to the gut. But in doing so, it turns into slightly different strain that gets favored. And then up here in the brain, as it moves from one part of the brain to the other, in different cells, the tendency to favor certain aggregates again comes into play. So in moving around in the brain, you may end up with several different variants of aggregate, and at least one of which is responsible for your disease type. So you have this strain cloud that arises from this particular aggregation starting in the gut and then being modified by different pathways through different cells, each affecting it according to their own conditions, and also how your personal genetics affects the conditions in the cells and what sort of things you may be exposed to environmentally. And the other thing that you see here is what ends out here in the periphery is not necessarily the same as what's in the brain where these diseases take place. We also know that viruses can infect the brain and, in fact, induce alpha-synuclein aggregation. So here we have brain first disease that starts with aggregation here at the number 1, but again spreads through several different cell types and then spreads out to the periphery. And so you end up with different aggregates in different, here's the skin, but also somewhat different in the heart, maybe something more like the skin and the muscle, something more like the heart and the kidney, something different still in the gut because it came down from the brain rather than started in the gut and went up to the brain. And so these authors have suggested that with these different types of body first versus brain first, you get these different strain clouds, and the cloud you get also is related to the disease, which again is going to get from the initial trigger aggregation through different cells focused down to a pathologic strain in these strain clouds. And depending on how that process goes, depending on your genetics, depending on what you've been exposed to, depending on what triggered the initial aggregation, you may end up with a Parkinson's-like strain cloud, or a dementia with Lewy bodies type strain cloud, or a pure autonomic failure strain cloud, or brain first, they think would most likely give rise to a Parkinson's disease strain cloud. So these two people would have very similar disease. There are some differences suggested that might come from where exactly things originated, but you would say that they both have Parkinson's disease. But then if you go and do a skin biopsy and a gut biopsy, you might get these two types of strains here and these two types of strains in the gut and skin there, and different arrangement of things, gut and skin there. And you do a biopsy of skin and a biopsy in the gut here, you get something different still. And the idea is that by using, again, these fancy fluorescent probes that can tell you how the alpha-synuclein in the aggregate is misfolding, you may be able to determine exactly what these are. And we may eventually be able to connect a fingerprint of what you find in the periphery to a particular strain cloud and a particular diagnosis. But it's not as simple as we had hoped of simply finding, oh yeah, whether you look in the skin or the gut, you either get Parkinson's disease strains of aggregation or you get MSA strains of aggregation and so forth. And then I keep talking again about these environmental exposures and genes, which I mentioned earlier. So this is just a couple of different genes and a couple of different forms of these two genes that could affect the risk of getting multiple system atrophy, but they also interact with environmental exposures. Now, this slide was made up so that everything ends up with a reduced risk of multiple system atrophy, which would be nice. But it's actually gets confusing because there's a lot of non-exposure and no this and so forth. So if we take the first one, this particular gene that's associated with oxidation, you can have various forms of it, and one of the forms has been labeled TT. And if you have this form of that gene, lots of variations out there in different people, and you're exposed to particularly nicotine or at least smoking, then maybe that can lead to an anti-aggregation effect on the alpha-synuclein by the nicotine. Of course, smoking's not good for you no matter what genes you've got, and it's not going to be helpful unless you happen to have this particular variant of this gene. But if you did, then that gene would help protect you from the tendency of smoking to, or from the possibility of triggering alpha-synuclein aggregation and result in a reduced risk of multiple system atrophy. So another form of this oxidation-related gene is the TC down here. And if you have this form of the gene, which some people do, many people don't, and you're exposed to certain pesticides, and we're going to ignore the non here. If you are exposed to the pesticide, then instead of all of these no's, you can end up with mitochondrial dysbiosis and reduction in ATP production, an increase in reactive oxidated species, and these can result in more aggregation. So this gene, combined with the appropriate pesticide exposure, actually increases the risk of multiple system atrophy. So if you wanted to reduce and you had this gene, you'd have to avoid the pesticide exposure, hence the non. Similarly, this is the alpha-synuclein gene itself, and it has variations. And some variations in the gene, which not everybody has, and exposure, again, we'll ignore the non and talk about exposure to well water that might have heavy metals in it. This can lead to alpha-synuclein aggregation induced by the heavy metals, the combination of well water with heavy metals in it and this particular variation in alpha-synuclein. And obviously, if it increases the risk because you were exposed, then you get more aggregation, you have more risk of developing multiple system atrophy or some other synucleinopathy. And similarly, a different variation in the gene is related to alcohol exposure. And if you have this form and you have alcohol exposure, and we ignore the non and the no, the alcohol-induced increase in serum levels of alpha-synuclein may contribute to the potential of developing triggering a seed of alpha-synuclein aggregation and getting multiple system atrophy or some other alpha-synucleinopathy. So it all comes together. The result is the tests that are available today can tell from spinal fluid or from a skin biopsy how likely it is that you have any of the synucleinopathies, but it can't tell us much about which one you've got. And the available tests have not been shown to differentiate between synucleinopathies. So you can't tell Parkinson's from multiple system atrophy, from dementia with Lewy bodies, and so forth. The research suggests that this differentiation of the alpha-synuclein aggregates and strains may be complicated, since multiple types of aggregates can be present in peripheral tissue samples and possibly cerebral spinal fluid, so that we may need a fingerprint of the different peripheral aggregates in different cell types to diagnose a specific CNS disease represented by a cloud of misfolded aggregated alpha-synuclein present in the brain and throughout the body. And this concept of strain selection gives us new insights into how the genetic and possibly the environmental factors can influence the risk of different diseases, different synucleinopathies by altering the cellular environment and the selection of which strains, when synuclein misfolding and aggregation is triggered, are propagated into the brain, the central nervous system, as well as to other parts of the body. And again, what strains or forms of abnormally folded aggregates may show up in other parts of the body, coming from the brain or from one part of the body to another. So life is much more complicated, but it's giving us a lot more insights into how these diseases work and get triggered. But we're going to need to do more research, which is actively being pursued, to actually have a test that we can look at some biopsy samples or body fluids and say what type of disease you actually have. So on that note, thank you for your attention.